All right, so today we have Dr. Camilla Bell. She received her master's degree in medical biology at the University of Lausanne. Is that how you say it?
Lausanne, yeah.
Lausanne, in 2015. And she received her PhD in Immunology and Molecular Pathogenesis in 2019 from the Graduate Division of Biological and Biomedical Sciences at Emory University right here in Atlanta. During her thesis work, Dr. Bell studied the mechanics leading to neutrophil plasticity in chronic diseases, with a primary focus on lung inflammation in infants and adults affected by cystic fibrosis. And she continued working on the mechanics regulating airways inflammation upon infection, including SARS-CoV-2, during her postdoctoral training at the University of Alabama at Birmingham. And currently, Dr. Bell is an assistant professor in the Department of Pathology at UAB, where her work focuses on the regulation of inflammatory responses in the cardiopulmonary system. And today, she will be talking about how we can leverage spatial transcriptomic analysis from patients affected by COVID-19 to inform mechanistic studies addressing the regulation of immune responses during SARS-CoV-2 infection. And with that, I'm going to hand it over to Dr. Bell.
Thank you, Hector. Thank you all for having me over for this seminar. So as Hector mentioned, today I thought to present some of the work and some of the stories that actually we worked on during the pandemic and a little bit after that. So with that, I would like to get started. Let me see. Okay. So some of these studies, we really designed it based on the fact that we had obviously a huge patient population that was affected by COVID-19 and no good animal model to actually study the pathology. So when 2020 started, we got finally access to some autopsy samples from patients that were infected with COVID-19. And so we decided to investigate certain aspects of the inflammatory response using a platform called NanoString. So the rationale behind the first part of the talk was that there were some reports, earlier reports, that SARS-CoV-2 had really good established link to thrombotic and cardiovascular events. And some endothelial cell infection actually was proposed to be one of the initiators of these events. However, there has been large controversy regarding this topic. And also the significance of the myocardial infection remained unclear. So to address this, we decided to collect autopsy specimen from patients that were infected with COVID-19 and then unfortunately passed away, and then from organs that or hearts that from individuals that did not succumb from a viral infection and were not actually either infected with COVID-19 and did not pass away due to a cardiovascular event. So we collected the tissue, and as I mentioned, we analyzed it using the NanoString platform. So we looked at areas that were more general, so as a 360 overview of what was going on in the heart itself. And we also stained for markers that were basically addressing the localization of endothelial cells like CD31 and obviously whether or not there was virus present. And we also looked at areas where there was actually, we were able to detect SARS-CoV-2 in the heart itself. So within the COVID population or the COVID cohort that we had, we also looked at differences between areas that presented with the virus and areas that presented without viral proteins. So overall, we didn't find huge differences between the two cohorts that we were looking at. There was a clear dichotomy between the two groups. However, the differential expression of genes wasn't as a clear cut. There were several genes that were involved, obviously, that we know that were implicated with SARS-CoV-2, like ACE2 and furin, and obviously the S protein that were not present in our control, but they were there in our COVID-19 tissues. But overall, the differential expression of genes wasn't as different between the two. And that also translated into the areas that were positive or negative for the virus itself. So if we were to look at areas that were actually positive for SARS-CoV-2 in the heart, the main genes that were upregulated were inflammatory genes related to peptide presentation or antigen presentation, like the HLA types, and obviously antiviral gene like the IFIT2 gene that is presented here. Overall, what we saw that the major infected cell type appeared to be endothelial cells, but of capillary nature. However, there was limited differential expression in the larger vessels. So that prompted us actually to go back and look at the site of SARS-CoV-2 infection, which is the lung. And the rationale behind this was that ARDS, or acute respiratory distress syndrome, has been observed in several viral infections, not just by SARS-CoV-2, including H1N1 influenza. So both of these viruses can actually induce ARDS, and both types have been characterized by increased lung inflammation and increased disease-related morbidity and mortality. However, there was some nuances between the two different types of infection that related to extended hospitalization, especially in SARS-CoV-2 patients compared to H1N1, which may indicate that even though some clinical phenotypes were similar, the immune response that was happening in the lung may have been different. So to address this, what we did, we went back to our collaborator in autopsy and collected or selected tissues from individuals that had passed because of COVID-19. And we confirmed this with actual immunostaining. And from some individuals that predated the pandemic that actually were infected with H1N1, and we also confirmed this by immunostaining. And during the early phase of the pandemic, actually this was probably May or July of 2020, we found that we actually had one individual that was co-infected with both viruses. So we also included lung tissue from this patient. And then in order to get a sense of what was happening in the lung, we selected different regions to get the transcriptome from. So we selected general areas, and I will call them general areas, and this will be basically just a mixture of cells. So we didn't go by cell type, but just a whole mix in areas where we actually could see presence of hyaline membranes. You can see them here in orange, which are characteristic of ARDS. We also then looked into the vascular bed that was next to these regions to see whether we could actually detect more meaningful differences compared to what we saw in the heart. And we looked, there is a type of disease, say epithelial cells in the lung, and at the resident and inflammatory macrophages that were present. Now, at the time of this study, unfortunately, the full transcriptome atlas by NanoString was not commercially available. So we used a smaller version of it, which included only 1,800 genes. So overall, we wanted to see whether there was obviously a substantial difference between the transcriptional profile in SARS-CoV-2 versus H1N1, and whether the double infected individual would actually resemble either more the H1N1 or the SARS-CoV-2 group, or if it would be like a unique profile on its own. So we looked at the general areas, and we also explored whether there was any difference in areas where we could detect the virus, or areas where we could not detect the virus. So overall, what we found was that SARS-CoV-2 had a unique profile compared to H1N1, and that in our case, obviously this is the N01, so take it with a grain of salt. In our case, this patient was more similar to our H1N1 group than the SARS-CoV-2. Now, this could have been, affected by the timing of the two different infections, or other factors that they're hard to tease apart with just one patient. But we thought it would be cool to actually include it in the same paper. And to address the comparison between infection and non-infection in our hands, we couldn't detect any meaningful differences between the two areas, suggesting that once the immune response gets going, the presence or absence of the virus in the tissue in that specific area, at least in our hands, didn't affect the transcriptional profile. So looking at general areas, we then explore which genes were actually differentially expressed. And as I said, the double infected individual wasn't much different from the H1N1 group. However, we could detect meaningful differences between SARS-CoV-2 and H1N1 samples. And these were related to epithelial to mesenchymal transition, extracellular matrix organization, including collagen genes that were highly upregulated in our SARS-CoV-2 group, and then coagulation and platelet activation, which would relate back to the thrombotic events that have been described in COVID-19. So in order to see whether we could actually detect more differences in the lung vasculature compared to the heart vasculature in COVID-19, we went on and looked at the vascular bed in these three different groups. And not many genes, again, were differentially expressed, but with those that were actually expressed differentially, you could see that there is a procoagulatory profile in SARS-CoV-2 that was not present in H1N1. So we then moved onto the epithelial part, and to see whether we could actually detect a differential activation of the resident cells, which are epithelial cells. And in this case, similar to what we saw in the general areas, we couldn't detect much difference between the double infected and the H1N1 group, but there were substantial differences between H1N1 and SARS-CoV-2. And here again, we could see enrichment of genes that are implicated in EMT, extracellular matrix organization, and motility and cell migration. So we also observed something very peculiar that was present in our SARS-CoV-2 tissues, but it was not present in our H1N1 cohort. And this was basically a hyperproliferation of epithelial cells in the lung that would form these almost like tumor-like clusters. So we were interested to see whether there was a specific profile or program that would define this hyperproliferation of the epithelium in SARS-CoV-2. And this was later on also described by other investigators with a completely separate cohort. So we selected areas where we could find this hyperproliferative profile of the epithelium and compared it to basically a normal epithelial structure. And as you can see, we could find small differences between the two. But one really interesting gene was actually the upregulation of KRAS, especially in the COVID cohort compared to the H1N1. However, we didn't have enough hands to follow up on this, but we know that it's something that could be explore down the road, and also with what relates to development of long COVID, especially with pulmonary outcomes. So the last cell type that we were interested in was the alveolar and resident macrophages, and some of the recruited inflammatory macrophages as well. So we stained for them and did the same comparison. And basically what we could find was that SARS-CoV-2 had a much larger enrichment for genes that are related to a M2 phenotype. And if you're not very familiar with the M2 phenotype, is usually more related to tissue remodeling and resolution of inflammation, or less inflammatory phenotype compared to M1 macrophage phenotype, which could actually relate to the fact that we see more collagen deposition in the lung in SARS-CoV-2 compared to H1N1. So to conclude this part of the talk, basically what we found was the SARS-CoV-2 had a unique profile that was related to the changes in extracellular matrix organization. There was a signature of epithelial to mesenchymal transition, so coagulation pathways that were upregulated, and then macrophages were more like an M2 phenotype compared to a more M1 phenotype that was found in the H1N1 group. So this takes me to the second part of the talk, where we actually took this information and went back to the bench. And so we decided to design a study to actually address, or see if we could address the evolution of the macrophage adaptation in the lung in SARS-CoV-2. So for this, we designed a study where we identified individuals that were actually infected with SARS-CoV-2. And out of those, the severe cases that were actually admitted in the ICU, we collected blood and tracheal aspirate at the time of admission and 5 to 7 days later. And then we performed flow cytometry, mass cytometry, RNA-seq, and followed up with some in vitro studies. So the first thing that we addressed was whether there were any changes overall in terms of the percentage of immune cells in these patients. So we collected samples from 52 patients at both time points, and phenotype basically the major cell types like T cells, B cells, NK cells, monocytes, neutrophils, and eosinophils. And as you can see, there is a huge neutrophilia that was described previously as well in these patients. But the percentages over time, at least within a week, they wouldn't budge as much. We also phenotyped these cells by flow cytometry, looking for differences in the macrophage activation phenotype and with relation to clinical parameters. So whether they were intubated for longer, or whether there was a correlation with their survival. And actually, we couldn't find a clear-cut outcome that would allow us to differentiate between patients that would go on and resolve the inflammation and those that would actually succumb from it. But at the time, as I mentioned, there was evidence that neutrophil to lymphocyte ratio in the blood was actually a very good biomarker for disease progression. And this has been shown in many other diseases, but the actual biological reason for it is still very a gray area, so it's not fully defined. And then presence of systemic neutrophil activation at the time of hospitalization actually related to most severe COVID-19 cases. And basically, there was one prior study that showed that increased neutrophil in the lung actually could obviously relate to ARDS, but the role itself remained unknown. So what we started with was to basically see if just by percentages we could discern between whether the patient would survive or not. So as you can see, there was basically no difference between the groups at either at the time of admission or at the second time point that we collected. We also looked for surface markers that could predict the activation of these cells. And as you can see, the overall neutrophil population, there wasn't a clear-cut marker that would allow us to discern between these two groups. And not just at the individual marker, but as a whole, based on what we decided to stain with for flow cytometry, we couldn't find a clear dichotomy between these two groups. However, interestingly, we found that there were different neutrophil subsets in the lung. And from now and through the rest of the talk, I will call these A1 neutrophils and call these A2 neutrophils. So A1 neutrophils are defined by a high expression of CD16, which is a Fc gamma receptor, and low levels of surface expression of CD63, which relates to granule release. And the A2 neutrophils start to lose expression of CD16 and actually increase CD63 at the surface. And we find these neutrophils not just in COVID, we find in non-viral ARDS, we find them in cystic fibrosis, we find them in H1N1 infected patients as well. So we don't know what they do in the lung. But our first question was, is it related to any clinical outcome at all? So what we found was that the percentage of A2 neutrophils actually was able to discern between those patients that actually did not survive versus those that stayed alive after ICU admission. And statistically, if you were to put a cutoff at 42% presence of these A2 neutrophils, there is a very clear difference whether you could survive or not upon being admitted in the ICU. So if you had low amount of A2, you were much more likely to not survive the disease. So for those of you that are not very familiar with neutrophil biology itself, so neutrophils develop in the bone marrow over the course of about 2 weeks. And over time, they start looking like a very normal every other cell type in the body. And they start making effector proteins that are then packaged into granules. So excuse me, they start with making proteins that are going to be packed in what they are called primary or azurophilic granules. And then these proteins include neutrophil elastase, myeloperoxidase, proteinase-3, cathepsin gene, and arginase 1, which are very potent enzymes. And then while they move through their development, they start to pack secondary granule, tertiary granule, then a set of secretory vesicles. And when they get activated, they start to release these mediators in a reverse order. So the first thing that they will release and come up at the membrane will be the secretory vesicles, then tertiary, secondary, and it's very rare that they actually secrete primary granules unless you are in a pathological setting. So for a long time, it was thought that if you were able to see these proteins in the extracellular milieu, it would just be because the neutrophils were dying off and releasing them. However, some of our work and others' work have shown that neutrophil can actually release these mediators and still be alive and perform other functions. So our body produces about 10 to the 9 neutrophil per kilo per day. They're typically short-lived in the blood. They live between 8 hours and a few days, although it's still very debated. They comprise 40 to 60% of all circulating white blood cells. And actually their aging profile is controlled by the microbiome. and their activation is dependent on the circadian clock in the body. In the lung itself, neutrophils actually tend to sequester in the lung. And the lung itself is thought to be a site of what is called neutrophil margination. And it's still debated whether it's a site where they get deactivated or deprimed, or is an actual immunological niche where they can interact with other cell types for different function. However, neutrophilia is very characteristic of many lung diseases, either due to infection or actual injury. And there are three main neutrophil phenotype at the macroscopic level. If you go and do single cell RNA-seq, you'll probably find more flavors than just these three. But for the purpose of this talk, we'll focus on the A1, what we call A1 and A2. So the primary granules that contain neutrophil elastics can be allocated into three different compartments. Either they can be fused with a phagolysosome, which gives rise to the canonical neutrophil phenotype, that phagocytose. And this is being defined by high CD16 expression and low CD63. Either the second phenotype is when the primary granule actually fused with the nucleus and promote a decondensation of the chromatin, and this turns into a process called netosis, where neutrophils actually spit out their DNA to capture bacteria, fungi, but also viruses, and proteins that are associated with the DNA can actually kill these microorganisms. The third phenotype that I was describing is when the primary granules are actually exocytosed. And these come with a transcriptional adaptation as well of these cells that seems to be related to their function. So in order to be able to basically manipulate these cells and study the mechanism, we couldn't just use patient sample. So we used an in vitro model that would allow us to isolate blood neutrophils and then transmigrate it through a collagen and epithelial layer, either towards a chemoattractant control like LTB4, and basically which would give rise to what we call the A1 neutrophils, or to use the supernatant from patients that were diagnosed with ARDS. So this supernatant is basically the tracheal aspirate that was devoid of cells and any of the bacteria. And these would give rise to actually A2 neutrophils. And then we isolated the cells and performed bulk RNA-seq on these. And what we found was that there was an increased expression or increased enrichment of genes that related to the interferon pathway and antiviral responses. And to make sure that our model was actually valid, we went back to a single cell data set that was actually finally published by another group. And we were able to actually find these A1 and A2 neutrophils in their data sets from patients with COVID-19. And as you can see here, the A2 neutrophils actually had increased expression of genes that were related to antiviral pathways. Now, the fact that we were able to access this data set is not trivial, since neutrophils are very fragile and many data sets that you see at the single cell level don't contain neutrophils, even though they are sometimes probably the most abundant population in the environment. However, this group was able to actually have a sizable population of neutrophils in their data set. So we then went in and basically made sure that the type 1 interferon pathway was actually enriched at each individual gene level. And as you can see here, the A2 neutrophils are very enriched for these genes that translate the type 1 interference signaling. And this was also true in the in vivo data set, single cell data set that that was publicly available. So we then looked into genes that were known or that were shown to be implicated in SARS-CoV-2 entry in the cells, replication, and late phase and release. And we saw upregulation of these genes in our A2 population compared to the A1, both in vitro and in vivo. So the next question is, are these genes up? Are neutrophils actually taking up SARS-CoV-2? So we performed image cytometry on patient samples that actually had high A1 percentages or high A2 percentages. And just as a reminder, these high A2 patients were actually, quote unquote, protected, while the high A1 were more susceptible to not make it out of the ICU. And as you can see here, we stained for SARS-CoV-2 for cholera-toxin B, which is actually a really good marker for neutrophils after fixation, and for CD63, which marks the primary granules. And as you can see here, patient that had high percentage of A1 neutrophils in their airways had, first of all, a higher amount of SARS-CoV-2, but higher amount also in the neutrophil population itself, and is quantified right here. So we went back to the actual supernatant of the tracheal aspirate to see whether we could detect any SARS-CoV-2 copies in there. And as you can see here, those patients that had high A1, so high virus intracellularly in the neutrophil population, also had higher viral loads in the extracellular environment. So this prompted us to see whether there was any difference on how A1, A2 neutrophils actually interacted with the virus itself. So in order to study that, we went back to our in vitro model and we transmigrated our cells, and then we incubated them with SARS-CoV-2. And basically, we then removed the extracellular virus. So the first question is, are they picking up the virus in a differential way? And that's why we see more virus in the A1 versus the A2. And we didn't see basically any meaningful differences, meaning that suggesting that they're basically picking up SARS-CoV-2 at the same rate. We then incubated the cells for another 24 hours, and we either took the cells and did PCR and checked for intracellular SARS-CoV-2, or we took the supernatant and basically incubated it on viro cells to see the extracellular amount of SARS-CoV-2. We also looked at viral replication, and there was no difference between the two subsets of neutrophils. However, interestingly, what we saw is that A2 neutrophils had less infectious virus intracellularly, and there was less infectious virus in the extracellular environment, which was really interesting. So we went back to our RNA-seq data and basically investigated whether the fact that these A2 neutrophils were able to have less infectious virus around was related to type 1 interferon signaling. So we did the same thing. However, at this stage here, we blocked type 1 interferon with an anti-IFNAR antibody that is available in the clinic called anifrolumab. And we had our IgG control. And as you can see here, with the IgG control, there was no difference in the amount of virus that was exocytosed. However, when you blocked type 1 interferon, you had increased amount of infectious virus in the extracellular milieu, suggesting that neutrophil may actually have a pathway for viral clearance that is related to type 1 interferon. So we went then downstream of that, and we went back to our RNA-seq data. And we found that there was one differential gene that was expressed when we blocked type 1 interferon, and that it was IFID3. So the role of IFID3 in viral infection is not fully understood. As of now, it's thought to be related to the degradation of viral RNA, but it's also related to activation of downstream proteins such as IRF3 and 7. So in our data set, we could see that IFID3 was highly increased in the A2 population. And I'm sorry that for some reason the stats did not come up here, but was significantly increased in our A2 population compared to A1. There wasn't very modulated when we added IgG, but it was highly downregulated when we blocked the type 1 interferon pathway. And in addition to that, there were genes that were related to RNA catabolism and protein catabolism and intracellular transport that were downregulated upon blockade of type 1 interferon, suggesting that between IFID3 and these other genes, there may be some relationship with how neutrophil actually deal with SARS-CoV-2 intracellularly. So the next step was to block IFID3 and see whether it would actually do anything in these cells. So we basically use a siRNA approach to downregulate IFID3 in our A2 neutrophils. And as you can see here, we can detect IFID3 expression in our negative control, but not just IFID3, but also a downstream protein that will be phosphorylated if IFID3 gets activated, which is phospho-IRS3. So once we block IFID3 expression by siRNA, we were able to downregulate dramatically both expressions and also the phosphorylation of IRF3. But at the functional level, we were actually intrigued to see that our hypothesis was true. And so when we blocked IFID3, we were basically preventing the A2 neutrophil from clearing SARS-CoV-2. And this was both at the infectious level of the virus itself, but also at the level of the RNA of the S protein and the N1 protein. So to conclude this part of the talk, we started with macrophages, but we then pivoted into the neutrophil population from the spatial transcriptomic studies that we did. And we were able to identify two populations of airway neutrophils, one termed A1 and one termed A2, that had differential expression of or activation of the type 1 interferon pathway, but also a functional adaptation related to that was basically SARS-CoV-2 clearance. And not only at the biological level, but there was a clinical basically outcome that was related to this, where low A2 neutrophils actually related to increased mortality in our patient population versus the high A2 neutrophils. Percentages seem to be protected. And we're currently having some preliminary data that presence of these cells may or may not related to the development of pulmonary aspect of long COVID. So we have a current single cell data set paired with transbronchial biopsies of patients with long COVID that we are analyzing. So maybe one of the next sessions, I'll be happy to present that. So with that, there were a lot of people involved in these projects. The team was great, and it was a huge collaborative effort, especially with the challenges of the pandemic itself and the limitations of it. And with that, I'll be happy to take any questions.
Thank you, Camilla. This was a great presentation. So, and it was a unique integration of non-spatial and spatial approaches mechanistically. I think that was very unique to your talk. So are there any questions from the audience? Please feel free to unmute or post on the chat, please. Sunil, do you have a question?
Yeah, I mean, I can always ask something. So I'm very intrigued with this NETosis. There's a lot of data about NETosis being important as a mechanism for protection of cancer cells. and development of metastasis.
Mm-hmm.
Did you identify any tools that one could use in the lab or something that can be taken further? I know I'm going off your SARS-CoV,
No, it's fine. Yeah,
but
yeah.
I'm a cancer guy.
Yeah. So there are ways of making neutrophil netos in vitro. And there are ways of quantifying the amount of netosis. Now, there is always going to be the question whether is it truly netosis or is just degranulation and death, and then they pair together. However, if the end product or the end result is the same, does it truly matter how they're formed in the first place, right? So at the cancer level, I mean, the idea is that, you know, the DNA itself provides a scaffold to carry proteases that then form the new niche, right, for the metastasis. So at the you know, experimental level, yes, you can make those in vitro, and you can, you know, mix them with your cancer cells and see, you know, whether in an in vivo model, if you transfer them, they produce metastasis or not. There are some inhibitors that, so there are different flavors of netosis too. It's not just one kind, but there are some inhibitors of netosis that have been used that showed some changes in the amount of metastasis. One of the enzyme that is required for a certain type of netosis is called PAD4. And there are some inhibitors that have been developed towards that. And I remember there was one presentation, I don't know if it ever got published, but that showed that inhibiting it with that PAD4 inhibitor in vivo would reduce the metastasis. You know, it was a 41 type of cancer.
41 model. Okay, cool. No questions there. So let me ask another one. If
Sure.
So in your lung studies, what type of pneumocytes are affected by SARS-CoV? Is it type 1, type 2? Are there bronchial cells? Where exactly is the primary damage? Were you able to discern it, or were the lungs too damaged to kind of figure those kind of things out?
So we use the autopsy specimen. So this is the very end stage disease, right? Based on others investigator studies, it seems to be an alveolar type of damage that occurs first. There are also different types of cell death that are imparted onto type 1 and type 2 in the alveoli that could, it seems to then result into a different inflammatory activation of the alveolar macrophages as well. So there is actually an investigator here, UAB, that is looking into that. And with the samples that we used, it wasn't possible to discern what it was, because it was so late in the disease that you can only do so much, right?
Absolutely.
Yeah. I mean, it would have been great to have, you know, access to transbronchial biopsies or something like that while they were intubated, but yeah. I mean, my hope is that at some point, you know, there have been huge advances in the development of animal models to actually study more mechanistically. So hopefully we'll get some answers from those.
No, I absolutely agree with you. The only, again, we were talking before your talk. We were discussing before your talk, I should say. The disadvantages are wise, and I think this is where, if you were looking at a larger region, you would have found some partially involved or early lesions. Again, it's autopsy, I understand. So just would be nice. Now, the virus particle, you didn't change the virus particles, did you?
I didn't what? Sorry, about the virus?
See where the virus particles were located? What they were in the fibrin, or they were in addition to the cells, or they were. What happens? I mean, where the cell gets destroyed, the virus is shed. Is it shed into the bloodstream? Is it shed into the airways? in the fibrinous exudate.
So I think
said
it's a
that I
little
missed
bit of both, and it depends on how severe your immune response is going to be to it as well. How much leakage you have from your blood vessel into the airways itself, and that would determine how much virus actually tracks back into the bloodstream. A huge question that is still not fully answered is how the virus actually gets back in the blood and gets into the heart and the vasculature in the heart itself. It's still very debated on how that happens, but I think the most straightforward answer to that would be, how much endothelial leakage you have in the lung, and basically how much virus you have that exited back into the bloodstream. There were some studies some investigators that proposed that, you have reverse transmigration of immune cells, and you could actually detect some viral RNA in the monocyte population in the blood.
Yeah.
But whether that's the mechanism that brings back the virus, or they're just going back to the bone marrow to re-educate the new wave of immune cells that is going to go back in the lung, is still not fully understood.
Great question. So as we are reading, and again, feel free to unmute, please. So I'm curious about your neutrophil phenotypes in situ. So you actually surveyed neutrophil biology, right, to a really great depth. I was wondering for spatial omics, where are we right now in terms of neutrophil profiles? Like, can we detect all these transitions in COVID tissues, or is it only certain phenotypes that you observe?
So I think you should be able to. So in our data set or in the limited data set that we had in that study, we actually saw a more sizable neutrophil population in the H1N1 group than in the COVID-19. It's possible that was, end stage disease, so it was a little bit different than while you're intubated in the ICU. But I think now with the development of single cell platforms, we finally got the cosmics here at UAB. So it will be really interesting to see at the single cell level. I'm sure you're going to be able to find it. There were some a study that was published, I think, right before ours, that they looked in blood. They didn't look in the airway, but even in blood, they were able to find different subsets of neutrophil, especially one cluster that had increased type 1 interferon expression or gene expression related to that pathway. Whether that population is what ends up in the lung, or is it primed through reverse transmigration and so develops over time, I don't think has been answered yet. But at the single cell level, if you have a good specimen, I think you should be able to detect them in the tissue and get the different RNA profiles from that.
Interesting. And then a relevant question is that you also showed that image stream with 3-color colocalization and after inhibition, what changes, right? I was wondering if you could, for instance, start with a fresh tissue, look at some of them in subsequent section with image stream, and then see basically isolating cells changes the distributions or not. There's a paper in Nature Methods about gene expression changes after you isolate from the environment, right? I was wondering if that's a biology that you'll observe or not. What do you think for the term?
So you're asking whether what I see on the image stream is going to be translated into the tissue itself?
Yeah, vice versa.
Yes, I believe so, because of the fact that we saw difference in the amount of virus in the two groups with a high and low A2. So biologically, I think there is a difference there. Now, whether it's to the same extent that or the same amount that we see on an image stream versus going back in situ, I think you're going to find differences there in terms of the level of it. But I think that it would be that difference would be present.
Interesting. So I think that would be interesting to see as well.
Yeah.
Great. I see that actually we lost half of our audience already, but
it's okay. We finished a little early.
I know. Are there any final questions? If not, let's thank Dr. Bell again. I'll use this clap sign, and thank you so much for your fantastic talk.
Thank you.
And then we learned a lot about COVID and spatial biology, as well as the non-spatial neutrophil biology that are synergistic. So with this, I think we have our next speaker in the agenda. Hector, could you tell us about the next speaker?
Yes, our next speaker on the agenda is coming up on April 5th. It is Yang Zhao. More information to become, but the title of their presentation is going to be Spatial Omics Mapping of Human Brain Disorders. So look out for that in 2 weeks.
And thank you, everyone. Again, happy Friday, and have a nice weekend.
Great.
Thanks. You too.
Bye.
Bye-bye.
Everyone.